All right. Hi, everyone. Welcome and thank you for attending Vector Builder's latest gene delivery seminar series. Our speaker today is Dr. Iuri Viotti Perisse, assistant professor at the Utah State University. After receiving his bachelor's degree from the University Federal de Viçosa in Brazil, Dr. Viotti Perisse completed his PhD at Utah State University, where he studied gene editing and genetic engineering and contributed to the development of cystic fibrosis and sickle cell disease models that more closely reflect the human condition. Ever since, Dr. Viotti Perisse has been continuing to utilize gene editing systems to create novel animal models for human disease and also for agricultural applications. So following the presentation today, we're going to have a Q&A session. So please type any questions that you have into the Q&A box at any time, and I'll read them out at the end. And for now, I'm going to hand things over to Iuri.
Thank you very much, Connie, and thank you everyone for attending the webinar. It's my pleasure to be here today presenting about the gene editing and all the stuff that we do here at Utah State University. So what I want to talk about to you today is about advancing gene editing strategies for efficient genetic improvements in sheep and cattle. In general, here we work with sheep, goats, and cattle, but we're not only limited for that. We also have some collaboration with small animals, but in general, we keep with these three species. But today I'm going to focus on sheep and cattle. Oh, let me see if I can. Oh, there you go. So I think the first question that we need to ask is, what kind of cell are we using to work? But that kind of cell will actually start with what's the purpose of the genetic modification we want to accomplish. So that could be a protein functional study, that could be a gene therapy development, or producing a model for a genetic disease that we want to study and understand better how that genetic disease works. Or we can even try to accomplish some improvements in agriculture in order to create some virus resistance in some specific species like sheep and cattle, or we want to introduce some traits like the pullness, which is removing the horn from bovine, and then you avoid the process of dehorning these animals. So these are some kind of things that we need to think about before we actually choose what kind of tool we're going to use, what kind of cell we're going to use, what's the best approach we're going to use to produce that kind of animal. Or if we just want to stay with the protein functional and study in vitro cells, how that protein works, what's the mechanism involved. If we want to just knock down some gene, we don't want to actually knock out the gene. So we have a very range of opportunities that we can do. So we're going to talk about some of them. So the strategy starts with what kind of cell you're targeting, what kind of tool you're going to use. Right now, we have a bunch of options. These options involve the CRISPR-Cas9, the CRISPR-Cas12a, Cas12c, Cas12a2. So there are many options. We're going to talk about some of them today. And we have also the Cas13. The Cas13 is a different kind because this one targets the RNA instead of the DNA. So it's a different one. But then we're going to define what kind of edits we actually want to accomplish. Do we want some random mutation in the DNA? Do we want to do some nucleotide replacement? And then we want to change just that little amino acid that is causing some disease. For example, in the sickle cell model that we have produced, and currently we are going to publish that one. That one is a single nucleotide replacement that caused a change in the amino acid, and that change caused the hemoglobin to polymerase and do all the effects associated with that sickle cell disease. So that's another option. The other option is a point mutation. Do you want to do a very small deletion? Do you want to do some small insertion in the DNA? So it really depends. And then I wanted to talk about what we defined. It was last year when I went to the International Society of Transgenic and Technology, and we defined as integrase editing as one tool that you can use either the CRISPR-Cas9 for integration of your transgene or using the passage that Dr. Liu's group generated. using primer editing for integration of that, or they evolved an engineered passage, which is another one they published recently, or using the base editing, which is a new molecule that is quite similar to the prime editing with small differences. And then we're going to talk about the animal production, which for sheep and bovine, we're trying to use more somatic cell nuclear transfer, and I'm going to explain why we would go with that, or microinjection, because microinjection is another simple way of accomplishing, but there are pros and cons with both methods. So let's go with the first step. Choose your right CRISPR toolbox. We have a range of different CRISPR Cas9, Cas12, Cas12A, Cas12C, Cas13, if you're targeting the RNA. So we know that the Cas now, since the discovery of the CRISPR-Cas pretty much 12 years ago, now we know many other small Cas that are associated with that mechanism of the immune system of bacteria. And we are going to focus on the Cas9, Cas12, and Cas13 here, because these are the ones that we can actually use to accomplish DNA and RNA targeting. So as you probably already know and are familiar with, this is the Cas9 structure. We have the gRNA tangled inside of the Cas9, and that gRNA actually binds to that double-strand DNA, where one strand will be the target and the other strand will be associated with the Cas9. And the Cas9 needs to have that PAM sequence, which is something that changes for every Cas, is having that specific sequence where the Cas will identify, which can be the NHEJ sequence, as in the wild type version of the Cas9, or in this new modified version with the NEG. So you need to have that NEG or NEGG downstream to your PAN sequence, because that's going to help your Cas9 to actually perform the cut, right? So once you have the alignment of the gRNA with the double-stranded DNA, the Cas9 have two main domains that will actually cut the DNA and cause the break, because that's what we want to accomplish, is causing the break, so the cell will be able to repair the break. The other option that is not very popular, but I have found to be extremely good in our lab, we have used this RNP version, is the Cas12a. The Cas12a is this protein here that you see in green. I can play that. Oh, no, I cannot, sorry. This protein here in green that is similar to the Cas9 in terms of getting that CR, the CRISPR RNA, and that CRISPR RNA will have the target, but it binds differently to the DNA. It identifies a different sequence of PAM, which usually, most of the time, it's going to be the TTTN or a TTN or a CTA. That depends on what kind of version you have of that Cas12. So once it binds there, it also cleaves the DNA, but it doesn't cleave the DNA in a blunt end, just like it happens with the Cas9. Here we have the Cas9 cutting the DNA and performing that double-stranded break in a blunt way. Here we have a stick end forming, and that stick end is also going to influence how you can actually fix that DNA. So another version of the Cas12 is the Cas12C. That one is not used for double-strand break, but it's used to knock down the gene. So you can use this version to bind to your target gene and avoiding that gene from being expressed. So instead of performing that knockout, you're actually performing a knockdown here, and you are avoiding the gene from being expressed. So you're not damaging the DNA, but you want to see what's the effect of that gene without that functional protein. The other one option that we have is the Cas13. The Cas13 is a different way because now you're not targeting the DNA anymore. You're targeting the mRNA that is being produced out of that DNA, right? And you don't want to, again, you don't want to do a knockout. You want to do a knockdown of the gene so you can understand the downstream that happens with that lack of the protein. So the Cas13, you're going to design a sgRNA where you're going to have your target. And once you have the target, it's going to degrade the DNA, the mRNA, and causing the absence of the protein. So you have all these tools that you can use for targeting DNA, targeting mRNA, and you're going to degrade. So what kind of actually edit you want to accomplish? Do you want to do one of those random mutation, just inserting any mutation that is going to disrupt the gene, or you want to do some more specific point mutations, some specific nucleotide replacement? So define your gene is also important because it depends on what's going to happen downstream in that application. So as you know, once we have either the Cas9 or the Cas12 targeting the DNA and performing that double strand break, the cell naturally is going to repair that mechanism of the DNA by using a non-homologous end joining or using a homology direct repair. Most of the time, the cell is going to use the non-homologous end joining by just ligating both ends using a bunch of proteins for identification. And once it identifies the break, it ligates that break. And that's where it causes random mutations to happen. So if you want to accomplish some random mutation, that's where you're going to go. So the homology direct repair is a more specific mechanism because you can drive how the break is going to be fixed. So you can use a donor DNA like a plasmid with homologous arms, which could be a huge homologous arms like 800, 1000 BP in each arm. Or you can use a small single-stranded oligonucleotide that is going to actually introduce that very tiny mutation that you actually want to accomplish. So you want to fix the DNA in that very specific way. And I'm going to demonstrate two very good examples with fairly high efficiency. So here's a mechanism that we have posed where it's more like a illustration that shows how a assassin in the end would find that target sequence that was broken by the CRISPR, and then it's fixed using that template. Another option for doing very precise genetic mutations is using base editing. The base editing is, they are doing different versions. So far, the most popular versions are the CBE and the ABE, where you have a cytidine deaminase or adenosine deaminase changing the cytosine here by removing that amine group, and then the cells identifies that as a uracil and fix that to be a thymine. The other option using the ABE with the adenosine base editor is removing that amine group, and then it changed to be identified as inosine. But the inosine is replaced by the DNA polymerase with a guanine. So what you're performing here, you are actually changing the cytosine to become the CG to become a TG. So it's a very precise single nucleotide that you are modifying. Another version that let me pass here. Another new version that you have for doing more precise and bigger edit. Of course, when I say bigger edit, it's not a huge insertion of the gene. And that one we're going to talk in a little bit about, but it's doing a couple of nucleotides. I have tried 38 BP insertion, so it's prime editing. The prime editing is basically the Cas9 that was modified to be an Integrase, just cutting one strand of the DNA. And then you have this new gRNA that was modified, and it's called prime editing gRNA. And that one contains a sequence that will bind to the opposite strand that was cut by the Cas9. And then you have a reverse transcriptase bound to that Cas9 that is going to reproduce that gRNA in a form of DNA. So now you have these little flaps here in the 3' end, and it's going to be competing with that 5' end. But the cell is quite good on identifying the right way that it needs to be fixed. But if you have that 3' end being inserted, the cell needs to go and use the mismatch repair to identify which one is actually the best. So since the prime editing was discovered and actually produced by Dr. Liu's group, they were able to perform this kind of genetic mutation, but it's extremely inefficient. I remember that back in 2020, 2021, I tried to accomplish prime editing in our lab using sheep cells, but it was quite hard to find the edit. But they have modified the prime editing to be now what's called twin prime editor. It's a little different because now instead of having just one uh, PEG RNA, you have two PEG RNAs that are targeting the DNA sequence in approximately 30 BP away. It's fine to be a little bit bigger, but not smaller than that. And once you have that happening, you're going to have this kind of bubble forming because you have a strand that was produced by one side and another strand that was produced by the other side of the prime editor. And now the cell needs to define which way is actually the best way of fixing that, which I honestly don't know if we know how the cell performs that identification and define which one is actually the best. It's something that I actually need to look at and find if anyone described this mechanism. But now you have a more and better chance of accomplishing that prime editing modification using this new molecule. So let's go to a more practical example where we're going to have the CRISPR-Cas9 working, and that's a design that we did when we were producing the sheep model for cystic fibrosis. We produced the 3 models. One of them was a knockout, the first one back in 2018, if I'm not wrong, no, 16 to 18. And then we produced 2 more models. One is the F508del model, which is the most common mutations in CF patients. So that happens in roughly over 70% of the CF patients. And the other mutation is a G542X. That one is produce a premature stop codon in the exon 12, and that causes the mRNA decay and not producing the protein at all. But the F508del is it misses this phenylalanine here at this position in the exon 11. And because of that, the protein is not really able to fold in the right conformation. And that caused the loss of function of that protein. And most of the time, it's going to be discarded. Sometimes it's able to fairly fold it and go to the membrane, but the protein is still not really functional because of that. So here's an example that we're going to use for production of the gRNA using the Cas9 regular approach, where we're going to select what kind of PEM we want to target. And then here you have the best options that you can use. If I'm going to use for just knock out the cells, I would go with this with high on-target scores because we have a better chance of accomplishing a high mutation efficiency by using RNP. But in this case, where we want to introduce a F508del mutation, we need to find the one that is extremely close to where that phenylalanine is present, which here is a 34 score for the on-target and 33 for the off-target, which is not really great, but we need to test because it's the closest one, basically. And then comes what kind of material we're going to use, what kind of CRISPR material we actually will use to accomplish that genetic mutations for either random mutation or knockin mutation. There are 3 options. One is using plasmid, which we used back in 2016-17 before the gRNAs came with the Cas9 produced commercially. And the maximum efficiency I have seen in our lab was about 40% of knockouts. And that's the problem, because in order to accomplish a knockin, which is a more specific modification, you need to have a high knockout efficiency. And we're going to talk about that in a little bit. So that's an option. The other option is using a mRNA, where you can produce the gRNA and produce the mRNA for the Cas9. And the other option that I honestly prefer whenever it's possible is using the Cas9 as a ribonucleoprotein complex, or RNP complex. So you have the gRNA with the Cas9 that you combine right 10 minutes for 10 minutes, and then you can actually transfect into the cells. So nowadays, I think we have the options for the Cas9 and also for the Cas12. We might have some other options, But these are the major ones that if you want to do with the RNP, you can use those. For prime editing, we have done with a CRISPR vector, which use the prime editing vector. And we also have done in vitro transcription to produce the mRNA for the prime editor and the PEG RNA. And we have done that transfection to introduce the mutation. So, okay, you define what kind of CRISPR you have, you define what kind of CRISPR material you're going to use, if it's vector, if it's RNP, mRNA, and now you need to send that inside of the cells. And that sending inside of cells is a little bit tricky because depending on what kind of cell you're working with, that could be a little challenge and limited. But we have pretty much four options. We can use a transduction, just producing a lentiviral or adeno-associated virus for that is going to deliver that material. We can use a lipofaction or liposome way. The problem that I have seen with lipofaction is that it does transfer your CRISPR, your material to inside of the cell. The problem is it's highly cytotoxic for most of the time and compromise the cells in long term, which for us is what we do. We need to have a mechanism that is quite reliable for transfection of the cells or transduction, that we're going to have cells later and we can understand if we had off-target mutations, if we culture the cells for a longer period. And usually we do because we do single cell isolation for that. But again, we're going to talk about that in a little bit. Most of the time, we're going to use a electroporation method because although it kills a lot of cells, I know it kills a lot of cells, but the cells survive extremely well after transfection, and you have extremely high efficiency with that. For microinjection, it's more applicable to oocytes and embryos. It's not very applicable for very tiny cells, but it's a mechanism that we can use. And we do some microinjection of bovine embryos and sheep embryos here. So let's go to the whole mechanism by using electroporation. We have a Lonza 4D nuclear factor here. So if we want to accomplish that knockout on very specific knock-in, we can use CRISPR-Cas with a ssODN or single-strand oligonucleotide. We can produce that prime editor or base editor using a in vitro transcription for RNA, or you can use a plasmid so you can transfect those inside of the cells, and then inside of the cell will produce the protein and the gRNA, and then the PEG RNA in that case, and then work inside of the cell. So there are different mechanisms that you can actually use, right? And then you go do the transfection using that electropulse. It's going to push that DNA, the material, inside of the cells. And then you do the genotyping. You wait at least 24 hours for the cells to recover, to have all the mechanism happening. And then you check by PCR if that's the mutation that happened. If you're introducing, like, let's say 38 BP using the ssODN, you can use PCR to amplify if you have actually that piece happening, or you want to just check for knock-in, small knock-ins or small knockouts. And then you do electrophoresis to check, you do sequencing to check. So once you have that sequencing, you can use a software called TIDE or TIDER for identification of your knock-in or your knockout. So going with the knockout mutation, here I have an example with that gRNA that I showed you for the CFTR gene. In that software, you're going to put what's the gRNA that you used, what's the wild type sequence, and what's the DNA that you modified from that pool of cells. So here we have the chromatogram of the pool of cells that we use the PCR to amplify, and we can see that the peaks are quite perfect to the point where it's getting close to where the double-strand break happens. And if you see the efficiency, it's almost 80% of efficiency. That means 80% of my whole PCR is mutated. only about 17% of the PCR product is actually a wild type sequence. And that gives you a p-value for this sequence in red, which is below 0.2001, and also a R-square for 96%. So for a knock-in mutation, in this case, accomplishing that F508del mutation, which removes, if you see here in the gRNA, the F508 mutation is the removal of the CTT, which causes the loss of the phenylalanine codon. So we input the wild type sequence, the sequence with the genetic modification, and the chromatogram of the test that you did, basically the pool of cells that you got. So again, you're going to have that chromatogram nearly perfect to the point where you start to have mutations, and the software is able to identify that mutation. But whoops, what happened here? Where's my HDR, my knock-in mutation happening here? Well, something that I found out about titer is that when you have this kind of small deletion, the software is not really good in understanding that that mutation is more associated here because we have the wild type with no mutations, small insertions, and a couple of deletions that happen. So if you check here for 3 nucleotides deletion, we have almost 44% of 3 nucleotides deletion, which I suspect that it's more related to my CTT happening there. So I think that's why when I checked for this mutation here, my HDR didn't show me the precise knock-in because it's removing the nucleotides there. So my knock-in is very likely involved in this 44% here, which when we isolated the cells by doing single cell isolation, about 30 to 35% of the cells, they actually had a double strand mutation for the F508del. So it's very likely that these 3 nucleotides deletion are right here. Another option is with the G542X, which I mentioned before. It's another very common mutation in the CF patients in the North America that caused the glycine to become a stop codon. So if you see the codon here, GGA, now we have a TGA for that mutation. And this one is a little bit more interesting because if you see here in the gRNA, we select that gRNA, we have the wild type sequence, we have the reference sequence with that very single nucleotide replacement, and then we have the sample that we did knock-in. So if you observe in this sequence here, you're gonna see that we have plenty of multiple peaks downstream to where the DNA is cut. But right here, we have a double peak, and then we have a single peak, single peak, single peak, single peak. That's a very good indication that you have a very good knock-in happening. So when we put in the titer is able to provide a better feedback on that and shows that 26% of the edits is actually related to the knock-in. So the total efficiency was 70%, and almost 1/3, actually more than 1/3 of that is a knock-in mutation, which is extremely good. So if you see the knockout, the wild type mutation, the wild type version is actually 28% of the whole PCR product. So here we have 29% of knockout mutation happening, a little bit 4 here, 5 there, but 26% of that is related to my knock-in, which means that if we want to do a very good knock-in, we need to do a very good knockout. So that gRNA needs to be extremely good to insert 70 to 80%, and I have seen over 99% knockout efficiency, which is not common, but that happens. So now I want to talk to you about the Integrase editing, which is not an official name, but it's a name that we put to say we can do a integration of transgene into target locus by using the ATTP, ATTB recombinant site. So the principle is pretty much the same regardless if we're using a Cas9, a twin primer editor, or a base editing, or this base editor that was created by this group here. So essentially, you're going to use a Cas9 or twin or that paste to insert either a ATTP or ATTB sequence inside of your target genome. So that could be in a locus that you already know and you want to introduce some portion of protein, some amino acids, or if you want to introduce a huge transgene like Cas9 and EGFB here, like in this example, or and after that, sorry, you're going to actually introduce that transgene with a BXB1 integrase. Once you have that BXB1 integrase, the BXB is actually going to use that ATTB, ATTP, and put both together inside of the genome. So with this mechanism, by using either Cas9 with SSODN to introduce first that ATTP or ATTB sequence, or using a primer editor to introduce that ATTB or ATTP sequence, or using the base editing or the base editor to introduce that ATTB or ATTP site. Once you have that site integrated, you are going to accomplish the target integration by using the integrase with your target gene of interest. So once you have that, you combine. That's why I call this integrase editing, because regardless of how you are actually writing that first ATTP or ATTB, it's going to introduce that transgene, that target integration by using the XB approach, which is actually an excellent way of not using the homologous recombination for that. Because if we depend on the homologous recombination with those very long homologous arms, it takes a little while to generate in the lab, or you can use the vector builder for ordering that. So instead of using that mechanism of getting that very long arm, you can use a vector with that very tiny ATTP or ATTB sequence, which for ATTB, just like it happens in this example here for the paste, it's 38 BP that is inserted. If it's a ATTP sequence, it's actually a 50 base pair actually being inserted there. So once you have that, you're going to accomplish that target integration because you're going to use your gene of interest and that BXB integrase to get your transgene and put inside of where you want to work. So here's an example that we have accomplished using that approach where we have the ROSA 26 locus, and we have done in both sheep and cattle, and we introduced a CRISPR-Cas9 targeting this ROSA locus here, and then we use a SSODN containing 150 BP, nucleotides to insert that ATTB sequence right here, and then we are able to use primers to identify if that was actually efficient. And then we can amplify that PCR product using a forward or reverse primer with the original locus primers. And then you do a next transfection with that vector, gene of interest vector with the opposite ATTP sequence in this case, and a BXB1. And then that BXB1 is going to be expressed inside of the cell and help you to accomplish that target integration, which you can use a PCR product just by using a primer with the ROSA 26 locus or whatever locus you're using with that vector you're using. So once you have that PCR product, you sequence and you can actually see that it has been inserted in that pool of cells that you have. So the next step is if you have an antibiotic resistant marker right there, or if you have a GFP that is guiding you, you can use that antibiotic resistant marker to eliminate the cells that didn't absorb the transgene. And then you have now a more efficient cells proliferating with that transgene. It's been a little bit. Now we're going to talk about how we usually do the production of these animals, the gene edited animals, here at Utah State University. Most of the time we do by somatic cell nuclear transfer, like I mentioned, but we have also done some microinjections, which is not really the best way in our case. Dealing with sheep and more sheep is a little critical. It's very sensitive embryo, but cattle is more robust for doing that. So we have done some microinjection to test CRISPR-Cas9 efficiency in those embryos. We have also transfected with some plasmids for trying the integrase editing and trying random integration, but it's still not quite that yet. I think we still need to improve a lot of these technologies to do this microinjection in embryos that will be quite efficient. So the problem about that is if you're lucky and if you have just a very straightforward knockout approach that you're going to knock out some genes and it's highly efficient because you're using a RNP with extremely good DNA, you can produce a knockout embryo easily. And then you can actually do a biopsy for checking that, even though the biopsy may not catch everything precisely, because if you have different mutations, but in general, a knockout as the cells divide, you still have a knockout, right? But the biopsy may not really help you catching all the mutations that inserted. The other problem is when you have a mosaicism here, is when as the cell divide, that CRISPR, that DNA is not very efficient in introduce the mutation that you want, and then it does produce different mutations or keep some Y type sequence, and then you have that mosaicism. So that's why we don't really like to use that approach. We like to use the somatic cell nuclear transfer, which is basically the name for the cloning procedure, where you have your fibroblasts in culture, which could be the sheep fibroblast or the cattle fibroblast. In this case here, as an example, we have the sheep fibroblast. We use the CRISPR-Cas9 with ssODN to do those precise genetic modifications. And then we can use a dilution of the cells to isolate single colonies that we can actually characterize and identify if they have knockout mutations, if they have Y-type sequence, or if they have the knockin, and if they have a combination of them in both alleles in the genome. So once you identify the homozygous mutation cell colony, you can actually use them to transfer into a mature oocyte that was enucleated by the micromanipulator, and that's what we do. We remove the DNA from that oocyte, and then we add that cell into the perivitellin space, and we fuse the cell together with that oocyte cell. And now you have that nucleus getting into the cell where you're going to use a couple of chemicals to activate the embryo, to induce that calcium oscillation, and then you have the activation of that. So once you have that within 12 hours for sheep, we do transfer those embryos into the surrogate mothers that were prepared for that. The only limitation that we have about sheep is that sheep is a seasonal estrus animal, which means that we only can do the cloning procedure between October to January, at least here where we are in Utah. So that's a little limitation, but we have the rest of the year to optimize all the things related to the gene editing approach and everything else, produce cells, because that takes a very long time. So once we produce that and we are able to transfer, we just need to wait 5 months to get the animals, which for somatic cell nuclear transfer in sheep, the embryo transfer gets about roughly between 20 to 40% efficiency. It really depends on the cell background. That's why, again, going all the way back when I mentioned the cell type that we are using, we have even difference when we have fibroblast cultures that some fibroblasts are better than the others, and we don't have an answer why, but they are. And that's something that we can see when it comes downstream in the somatic cell nuclear transfer to produce those embryos. So we can have embryos that are extremely good, and we get like 40% of the pregnancy in those embryo transfer, or sometimes we don't have that very good embryo, and then we have like 20% of pregnancy that actually will go to term. And then we have the clones, right? So the advantage of using this method, we have quite a bit. First one is the ability that we have to verify all the genetic mutation. If it's a homozygous, it's a heterozygous mutation. And if you have off-targets happening, off-target is something that concerns quite a bit when we want to think about applications in agriculture, because these are applications that will downstream affect the consume of these animals, or I mean, if we're going to do genetic modification for them to use for human consumption very later in the future, we need to worry about if we have off targets happening. And by using somatic cell nuclear transfer, we can actually screen those cells and see with the background cell if we actually have some genetic modifications that were not introduced or they were introduced by CRISPR-Cas9. And also it eliminates the genetic mosaicism that happens with the microinjection. So we don't have the problem. All the cells will be exactly the same. All this animal here, and this is actually an image of the farm that we have with some cloned lambs. They are identical to each other. So it's extremely good way of producing animals. And this work I have been doing with Dr. Polejeva, who is an extraordinary researcher here at Utah State University. She has been involved in multiple models production, and I have been honored to work with her since my PhD. So we have produced many different models with human disease applications, or even for agriculture application. So these models includes the sickle cell model that we are going to publish now. We have all the CF models that have also been involved. We have the Xeno for the GGTA knockout sheep that is similar to the pig used for xenotransplantation. So now we have this model that is going to be used for xenotransplantation. We have the interferon model that is also helps with understanding how the viral infection happens in sheep. So many different models that we have produced here since I came to Utah State back in 2016. And I would like to acknowledge Vector Builder and the team for inviting me for this presentation, and acknowledge all the team that we have here at Utah State. So all this work is thanks to Dr. Liu, who is this Chinese lady here, Dr. Polejeva holding this little goat here. This one is me back in 1820, I don't remember. And all the crew that we have involved in this research, all the funding that we got through USDA, Utah Agriculture Experiment Station, and NIH, of course. They have been extremely helpful. Also acknowledge Dr. Ann Harris, who helped us characterizing the sheep model for cystic fibrosis. She's extremely knowledgeable in that area. And doctors Graça Almeida Parada and Chris Parada, because they are helping us producing the sickle cell model for this sheep model for sickle cell disease. So with that, I would like to thank everybody for being here in this presentation.
Fantastic. Thank you so much, Yuri. That was wonderful.
You're welcome.
We already have a couple of questions that are coming in. So the first one being, you introduced a lot of really useful information about the use of different Cas enzymes, and one that you mentioned was Cas13 for mRNA degradation. So do you know what the benefit is to using Cas13 versus using CRISPR-I with dCas9 as an approach?
So the CRISPR-I pretty much blocks the expression by literally sitting on the DNA. So that will be more similar to the Cas12c that binds to the DNA and prevents the expression. The Cas13 is more going towards the mRNA and cleaving that mRNA. I think both ways works well. It's possible that it's much easier to target the DNA because you have two alleles, so you target both at the same time. Then you actually target the mRNA that is more mRNA that we have for targeting. So I have not tested or tried to compare both ways, but if I would have an opinion, which way would be best, it could be by using a Cas12c, by sitting on that sequence and preventing the protein from being produced or the mRNA from being produced. than using the Cas13. But of course, Cas13 is also another option, and perhaps we can control which way will degrade more or less. With gRNA, we will degrade more or less the mRNA.
Perfect. Thank you.
You're
The
welcome.
next question is, have you considered using AAV to deliver the donor template, since this has been a quite popular method for mouse line engineering?
I have considered, but the problem is it takes a little while to produce the AAV for delivery. And for us, the electroporation is extremely efficient in getting the fibroblasts with the genetic modifications we want. So, there's something that we say in Brazil is when you have that team winning, you don't change the team because you don't change the players because you're winning, right? So it is something that we can consider, but I don't know if that's worth it. Probably depending on what kind of genetic mutations we want. For these small genetic mutations, it's super easy to do the electroporation with the ssODN to introduce the precise mutation. Perhaps it would be good for using a system like the integrase editing where we have that BXP and the gene of interest, but the problem is the size of the DNA of that plasmid that you can actually have. So
Hmm.
yes, for a gene like the CFTR, it's a huge gene with almost 5,000 BP. It's a little hard to play with that and use a AAV because of the size of the cargo that you can actually have.
Right, right, absolutely. We would be happy to help troubleshoot that if you're interested in trying out some AAVs.
Absolutely.
So the next question is, what are the off-target effects associated with Cas9, Cas12, and Cas13?
So it really depends on if you're using the plasmid, mRNA, or RNP. If you're using the RNP, it's very likely that it may happen some off targets to happen, but something that is also a advantage of using somatic cell nuclear transfer, let's say in a hypothetical example that once we do the transfection with Cas9 to introduce a mutation, and that Cas9 is extremely efficient, 99% efficient in doing a on-target mutation, right? But that problem is, let's say you have 10% off-target happening, which is a quite big amount of off-target, right? When we select the cells, we have 3 million cells. And then we need to dig into those 3 million cells to find the cells with the biallelic mutation that we have. And the likelihood that we're going to have that off-target also happening there is very low. It's not only that 10% is much lower because we are selecting one cell that has both mutations that you want in both alleles, and also you dig into the off-targets. So far, all these cells that we have produced here and we yet have used for somatic cell nuclear transfer, we have not identified that. And I think it's more because of probability, that the probability that you're going to identify a off target in a cell that you select from that single cell that you expanded into a population, and that single cell actually brought some off target mutation is very low. So we need to be more unlucky to get that off target mutation. But of course, all these Cas9, Cas12, as much that they get efficient, they also cause off targets for a big population. We we need to try to avoid and consider how much that off target actually matters. And that's actually why using a primary editing, and that's something that I have interest in moving on here, is using the primary editing to generate mutations that are associated for agriculture with agriculture application, like removing the horn of cattle. So you introduce that, and let's suppose I have some cells with off-target mutations, but then when I screen these cells that I'm going to use for doing the cloning procedure for animal production, that one does not have, because the likelihood that I'm getting that one with a off-target mutation is very low. And the prime editing is already very good in avoiding off-targets. So that's also why I have quite faith in using prime editing for moving on with these agriculture traits.
Perfect. That makes sense. Thank you.
Yeah, thank you.
So as a sort of final question, so it was great to see all of the different models that you've created for approaching various different diseases as well as agricultural problems. How would you go about selecting a particular model for a particular disease? So for instance, Crohn's disease is an ideal sort of mouse model. So how would you go about determining which would be the best model in terms of sheep or goat or something completely different?
That's a excellent question. For example, we have used goat to study to produce actually these goats that we have in this picture, where they are immunocompromised by not having their own immunoglobulin, and they have the human artificial chromosome to produce a human antibody. So we need to see what aspects in that animal that is more advantage. You can take more advantage in being more similar to human in the case of genetic disease. Another example is the cystic fibrosis and the sickle cell models. Why we're using sheep instead of a goat. So sheep has been studied for a long time, and it's very similar in terms of lung development. And lung disease in CF patient is something that affects a lot. So the lung is very similar to human. So that's why we actually use sheep as that model. And also the mechanisms that involves the production of the red blood cells in sheep is very similar to human, because when we are producing the animals, something that happens to us humans is getting a switch from the fetal hemoglobin to a adult hemoglobin. And that happens when it's very close to the birth. And that also happens in sheep. They also have that switch from the fetal hemoglobin to the adult. And then once, I think it's about like 2, 3 months after birth of these animals, they have the whole switch already happen. Everything already switched to the adult gene. So, but in human, that happens in within a year. So this mechanism is very similar. So that's how we actually choose and define what model we're going to use. And sheep has been extremely a good model. We also had some cardiac model using goats because also of these very specific mechanisms that are similar and the size. So if you think about sheep and goat, they grow to the size that it's very comparable to us, to humans, right? The size of the heart, the size of the lungs, and that's actually why we used a sheep as a model for xenotransplantation, because the size is very close to us. But for example, we're not going to use cattle for this kind of disease because they just get too big and it's not very close. So these small ruminants, they are much better for using. But you need to, of course, we have collaborations to study better all these diseases. We need to see which model actually will fit better in that case.
Perfect. Excellent. Thank you so much. That was a wonderful presentation and a really engaging Q&A session. And we really appreciate you taking the time and giving such a really comprehensive and informative seminar. Thank you so much.
A pleasure. I appreciate all the audience here and appreciate for the opportunity to talk about our work here. It's quite unique in the whole United States, to be honest.
Perfect. Yes. Wonderful. Well, thank you so much again. And thank you everyone for attending. And we look forward to seeing you next time. Thanks, everyone. Bye.